You guys are used to my recorder now with the countdown. Okay, so to get started, we're actually going to focus on 3 different windows today. So up in the top left-hand corner of your screen, you're going to notice we have 3 buttons labeled Arena, Surpass, and Vantage. Okay, so those are the windows we're going to be looking at during this session. And we're going to start off with the Arena window because when you start the application, it's the first window you're going to see when you open up the software. Okay? The Arena window is really here as a tool for data management so that you can bring your data in, visualize your data, organize your data, and also if you're interested in batch processing, this is where we organize all of the batch processes also. Okay? So for right now, I'm going to keep it really easy. We have a list over here on the left-hand side that basically shows you a folder organization, okay? You only have one folder or one group called Imaris Demo Images. You'll see I have quite a few more. So the idea is that you can organize your data using 2 different folders, okay? One we refer to as an assay, which is like a master folder, and one that we call a group, which is like a subfolder, okay? So in your Imaris Demo Images, if you were to open that up by clicking on it, you'll see that you actually have 2 groups inside of there. So you can organize the data based on your experiment, based on the microscope you acquired the data, or maybe you want to have a lab assay that inside of there you have multiple different users. So you can organize it however you choose. So I'm just going to show you to create an assay. We're going to click on that assay button. You'll notice it will come up and ask you for a name. I'm just going to call it test, which is original. It's going to add a little basket into that list you see on the left hand side with that name. That basket is currently empty. You'll notice your Imaris Demo Image basket has red blobs in it, meaning that it contains data, it contains an item. If you want to add a subfolder to your assay, you can click on the group icon and that will add a group into the assay that you have selected. Okay, so I'm going to call this Experiment 1 and click OK. So it now adds this square folder or square group into that assay. Okay. To get data in here, you're going to select where you want the data to go by either selecting an assay or a group, and then click on the image icon, which looks like a red blob. Okay. Click on that image icon and it opens up the open dialog. Now, I do want to point out one thing in here. There is a little pull-down list called Files of Type. In that list is a complete listing of all the different file formats that we support in Imaris. So if you're ever unsure whether we support a particular file format, you can see a complete listing in here. Okay? So if you want to open up data in a particular group or in an assay, you can then go in and select those files and it will bring them into the group for you. Okay? Now, what will happen, and I'm going to go back to my demo images folder, which is a little bit more comprehensive than yours, but you'll see that every single image will be represented with a thumbnail. And that thumbnail is basically a little maximum projection, maximum intensity projection of the data within that file. Okay? You'll also be able to put your cursor on each of those previews and it will give you a breakdown of how many channels is in that data set, what the voxel size was, where that data is located. So you get a breakdown of the information on the data set itself. Okay? Now, we're going to start off looking at a particular file. So if everyone could browse to the thumbnail of the retina image, red and green retina, hopefully you guys see that. So in order to open that and start working with a 3D volume, you're going to double click and that will launch it into the Imaris Surpass view. Okay, so you'll notice up in the top left hand corner. Okay, so go back to Arena and then click on here on the Imaris Demo Images. That's right. Okay. So that's going to open up the file into the Surpass view. Now the Surpass view in Imaris is your main work area. That's where you're going to do your visualization, all of your analysis. It's all going to be done within this view. I do also want to show you, for those of you that don't want to organize your data in Imaris, that are not planning to do any batch processing and just want to open up an image whenever you want, you can go straight to the Surpass view. So it will open up an arena, just click on the Surpass button, it will take you straight here. Then you go to the File menu and click Open, and you can browse to wherever your file is located. Alternatively, you can drag and drop a file right into the Surpass view. Okay? So I just want to explain that there's multiple ways to get data into Imaris, but if you want to just go straight to the Surpass, you can drag and drop and do that. So this morning we talked a little bit about 3D visualization techniques, so that's where we're going to get started. So when you bring your volume in, how can you change and look in different rendering modes? So you'll see over on the left-hand side that you have a list of objects. This is what we call the Surpass scene list over here on the left. Okay? These are 3 objects that get created every time you open a data set in Imaris. So we have a light source, a frame, and a volume. The volume object is your image data. So you'll notice there's a little checkbox next to it. So if you turn that checkbox on and off, you'll see that the data will appear and disappear. Okay? So that is your image data. We also have a frame. So you'll see around the very edge of your image, you see a white box with a grid. That is your frame. And you'll see also if you turn that little checkbox on and off, it will allow you to turn the frame on and off. Now, down in the bottom left-hand corner, when you click on a particular object, you're going to see a configuration for that object. And in the frame, you can configure it to have a box, take the grid out, change the grid size. You can configure it however you would prefer. Now, this concept is consistent throughout Imaris. So when you click on an object in the bottom left-hand corner, you'll be able to configure that object. And the same is true for the volume object. Okay? The volume object, once you click on it, has the 4 different rendering modes that we talked about this morning. Okay? So by default, it opens up in a maximum intensity projection, but we discussed that that can make it look very flat and 2-dimensional. So I want to show you how we can take the other rendering modes and make the data look more 3D. So, just before we do that, one other dialogue. Everyone should have a little window that floats on top of the Imaris window. It's called the display adjustment. If you do not see it, it might have been closed, and you can reopen it by going under the edit menu, go to show display adjustment, and it will open it for you. Okay? Now your display adjustment controls your channels. Okay? Not only turning them on and off, but it also allows you to change the color of channels. It also allows you to change the display or the contrast. Okay? So you'll see on your display adjustment you have 2 channels. They're labeled based on the object that has been, or the fluorophore. And you'll see that each one has a checkbox, so you can turn them on and off independently. Oops, excuse me. We'll come to that in a second, but each one can be turned on and off. Each one also has 3 black triangles that allow you to change the display. So what that means is if I grab this top triangle on the right-hand side and drag it down, you'll see that it will increase my brightness. The bottom will change your background, and the one in the middle is your gamma control. So gamma is effectively an amplification of the intermediate intensity values. So if you have a lot of bright structures, but you also have a lot of dim structures, and you want to amplify the dim so you can see them more clearly, you're going to use the gamma control to help you amplify those intermediate without overdoing the bright. Okay? You're effectively adding curvature to your display. So the gamma, the brightness, and the background, all 3 of those change how the data is visualized, but the underlying data is exactly the same. So the intensity values are not changed. So this is purely a change for display, not intensity. Okay? So now that we know that we have controls for display.
Sorry, just for that, when you open it, does it automatically do kind of a best fit?
It does.
Okay.
It does a best. So it basically does an auto adjustment when you first open up your data, because the assumption is Most of the time, the data is at the bottom end of your scale because you don't want to photo bleach, so people tend to keep their laser lines low. So we'll automatically set an adjustment for display. You can then adjust that yourself to get it to where you want it to be, or there's an option in here to reset all channels, and that takes you back to the default lookup table. Okay. Now, the next thing I want to tell you, so currently we're in maximum intensity projection, and what I want to do is change into blend. So for those of you that were here this morning, blend is where we add opacity and shading in order to be able to visualize more of a 3D perspective of this data set. So if I click on the blend mode, a couple of things are going to happen. The first thing that's going to happen is the background will turn gray. Second thing is the data is going to look more solid. Okay? So when I say more solid, you're not going to be able to see through it like you were when we were looking at a maximum intensity projection. And hopefully instantly you should be able to tell that these green fibers are running underneath this vessel. So you're getting a three-dimensional perspective straight away. Now we can change and optimize this visualization, and this is going to be important for you when you're bringing in your own data. So I want to explain this little dialog, this blend mode control that you have in your volume object. So each channel has its own blend mode control. Okay? Each channel has control for opacity. So if I drag this point up and down, you'll see that I can change the opacity of a particular channel. So I'm in the red channel right now. I can switch over to the green and also change the opacity in the green channel. Okay? I can also change where these points are, and this is going to impact what you're visualizing. So this bottom point right here allows you to control what is visualized and what is not visualized. So anything below this point is set to 0 and turned off. So if you have high background, you can use this value to help eliminate the visualization of that background. Okay? Now, you can use this slide or this point at the top to drag left and right to make your structures or make dimmer structures look more solid or less solid. So moving backwards and forward is changing the intensity values that are visualized as an opaque structure. So we can combine the two together, and this is where you can optimize it for your own data set. So based on what you're trying to visualize, you can play with this blend mode controls in order to make it look as good as you can. Now, I also want to show you one other thing. So this morning we talked about a new feature that we have for the visualization in a blend mode called peak. Do you remember there was a hollow structure that we were looking at? So I want to show where that is accessed. So at the bottom of their blend mode dialog, there is a little option called graph shape. We're currently in step mode. Okay? There is another option called peak. And what I'm going to do here is I'm going to turn the green channel off in the display adjustment dialog, and I'm going to look at just the blood vessel on its own. Okay? What we've seen now is instead of having 2 points on this plot, we now have 3. So we basically have a little range. So everything below a certain value is turned off, and then above a certain value is turned off. And we're only visualizing the information in between these 2 points. Okay? If we drag these 2 points further apart, it's going to include more information. If we pull these 2 points together, what we're going to start seeing is that the structure that we're looking at becomes hollow. Okay? Now, we still have access to this opacity. So I'm going to bring this out a little bit. Oops. I should look at my screen, not the projector. Okay. You can bring this out a little bit, and what I'm going to do is I'm going to grab this middle peak and bring it down. And when you do that, you're basically setting the opacity level for that particular structure. So this is basically a way to visualize your data using a boundary. Okay? Thank you very much. So when you visualize data, remember I was saying when we were playing with the opacity in the step mode, the dimmer structures will become opaque before the brighter. So if you're trying to adjust the opacity, you lose your dimmer structures first. this technique allows you to specify a particular range that you're interested in, make them hollow so it's easier to see things inside. Okay? So, do you have a question? No? Oh, sorry. Okay. So, the peak mode is one that you can use for visualization. It makes it easier to display and also to present particular data sets as well. So if I go back to the step mode for now, I want everyone to know that when you go into one of the alternative rendering modes, you are going to have some other controls that you can use to optimize the visualization. Now, we also have another mode called normal shading. So this is the third of the 4 different rendering techniques. And if I switch that on, you're going to notice straight away it looks even more solid, and it typically looks even brighter than it did before. Now you have the same exact controls as you had previously, so you can drag the slider bar back and forth to adjust what intensities are being made solid. You also can adjust the opacity like we did before as well. Now the difference with this mode is we have an additional light source that's being incorporated into this reconstruction, and I want to show you where that is. So if you put your cursor on the 3D view, you'll notice you have a little cursor of 2 arrows. You're in navigate mode, which means that if you click and drag the data set, you can rotate it, and if you click, drag, and let go, it will rotate, okay, on its own. Click again to stop it. You can also zoom in and out using the scroll wheel on your mouse, okay? Now, I'm going to zoom out a little bit because I want to show you this object at the top of your scene list called light source. And this is something that if you click on, you're going to see a little representation of your light source. Okay? Looks like a little thin line with a plunger-like structure at the end. This is how we're going to control the orientation of light. So, you'll notice that if I try and access it or control it or drag it or do anything, it won't let me do anything in the mode I'm in. So there are 2 modes in Imaris for visual or for control of your 3D volume. One is navigate. You're currently in that with the 2 arrows. If you hold your escape or tap on your escape key, you'll notice that your mouse cursor will change into a pointer, and that puts you into a different mode called select. Okay? Select allows you to interact with objects in your 3D view. So instead of just rotating and zooming, now you can actually select and move and drag. Okay, so now that I'm in select mode, so just so everyone knows, the escape key is going to become your friend. When you're in Imaris, you'll have one hand on the escape key and the other hand on the mouse. Okay, so the escape key is basically a toggle, but now that you're in select mode, you can grab that light source and you can orient that light source in whichever direction you want. So bear in mind, the reason we do all of this is to make it easier for you to optimize your data and visualization of your data. So if there's a particular orientation that helps you emphasize a particular cell population or structures of interest, you can orient it in whichever direction you'd like. Okay? Also, you can put your light source behind your structure, which is not at all useful because it makes it look a lot darker. So just be aware that you want to try and keep your light source in front of your data rather than behind. Okay. The last rendering technique, so I'm going to click back on the volume object. The last rendering technique is called a shadow projection. And shadow projection is our most computationally intense, but it is the mode that allows you to cast shadows behind. So if you have structures that are sitting in front of other structures, it will obscure the ones behind. So if you click on shadow projection, you're going to notice that it will take a little bit longer to generate the image. First thing, there's usually a little progress bar in the bottom right-hand corner. Also, as you make changes, so maybe you want to change the display, or maybe you want to change the light source, you'll notice that as you do that, it will show you a progress bar in the bottom corner. So changes will require it to be rebuilt, and therefore it is a little slower than the other techniques. This technique is really here for those people who want to get a good snapshot for a poster or a publication. It's not really designed to be an interactive preview. So if you want interactivity, use one of the other modes that we provide. Okay? So I'm going to go back to maximum intensity projection, and I'm going to turn the other channel on. So hopefully that makes sense. So now you know where the other rendering controls are and you have access to how to optimize. The next thing that I want to go over with you, and I'm going to make my scale bar a little smaller. Sorry, it's kind of big. I want to go through a surface reconstruction, okay? So we talked about this morning. We talked about different rendering modes. We talked about modeling. The first model I want to show you is a surface model. So in order to create a surface object, we're going to have to add it into our scene. So to do that, I'm going to go to the top of that scene list, and you'll see a number of different icons. Some of those icons should look familiar because I used them in my talk this morning. If I click on the blue blob, that is our surface icon, and if you click on it, it will add a surface object into your list. It should be called Surface 1. You can double click on the name and call it whatever you want. Now I'm going to make we are going to make a surface reconstruction of that blood vessel in here. So I'm going to call this vessel. Okay. The more objects you create, the more likely you're going to want to name them so you remember what they are detecting. So you have a list of objects that are being detected, and you can easily tell which one is which based on the naming convention. So I'm going to call this one vessel, and you'll notice down in the bottom left-hand corner you have a configuration for the surface. This is actually what we refer to as a creation wizard, and a creation wizard is a set of steps that guide you through the parameters you need to choose to create the perfect object. Okay? So we're going to go through this creation wizard, and the very first step of a surface creation wizard is asking you whether or not you want to incorporate a region of interest. So a region of interest may be that you have a particular part of the image that has structures that you're interested in and you want to exclude everything else. You may also use a region of interest just to set up your parameters and then want to apply those parameters to everything after or at the end. So in this case, I'm going to go through and segment a region of interest. So I'm going to click on that checkbox. You'll notice when you click on that checkbox, there's another option that comes up right underneath called Process Entire Image Finally. So this is where you have the option of using it temporarily or using it as a region of interest that will carry through. Okay. In this example, I'm going to have you click on Process Entire Image Finally. Okay. We're then going to go down to the bottom where you'll see a little blue arrow, which is called forward. We then have a green button for finish and an orange button for cancel. So we're going to go to the blue forward arrow, and that'll take us to the next step. This is where we can adjust the region of interest. So hopefully you can all see a box with little arrows on each of the faces of that region of interest. Okay. So remember select mode. So if I click on the escape key, it puts me into that select mode. Here is where, if I switch into select mode, I can grab one of those arrows and adjust that region of interest. Okay? So this is where you're going to have one finger on the escape key, the other on your mouse, and you can reorient and grab that region of interest from different sides or different faces of that region of interest and move it around and size it based on what you are looking for. So I'm just going to use a region of interest just to show you how this works. So I'm just putting a region of interest that has some of the vessel in it, and I'm going to go to the next step. So the third step is asking you to choose the channel, and also it asks you to choose the thresholding technique. So I'm going to go through this method with you in the default mode, which is called absolute intensity. I'm then going to have you switch to a different data set, and we're going to go through the other mode, which is called background subtraction, because I want you to understand the difference between the two and when to choose one versus another. Now, we have one vessel, one continuous vessel in this data set, and for the most part, it's the same intensity. Okay? Absolute intensity allows you to choose one threshold value, that is the difference between signal and background. The background subtraction technique is more designed for data sets where you have variation in intensity. So if you have structures that are dim and structures that are bright, you're more likely to use background subtraction. So I want to show you how this would work. So we're going to leave this in the default, and go to the next step. And here we have a silver surface. And this is basically a little preview. This is our histogram of our intensity profile. And if I click on the edge of that yellow bar, you can adjust the threshold. Okay? So if I bring it down, it incorporates more. If I bring it up, it incorporates less. But you can see interactively a surface being displayed for that region of interest. Okay? Now, I'm going to go to the next step, and that is going to allow me to do some classification. Now, when I only have one structure, it's not terribly useful, so we're going to come back to this one with a different data set. So for now, I'm going to hit the Finish button, and that is going to allow us to build that surface for the entire volume. Okay? And so you should have something that looks a little bit like what I'm showing on the screen. Okay? Now, I'm going to turn the volume object off using the little checkbox beside the volume object, and that's going to show me just the surface on its own. And a couple things I want to show you. I should have shown you this a little bit earlier too, but you'll notice inside this vessel there are some little lumps and bumps in there. You can see the kind of morphology of the inside and the outside of the vessel. Surface makes it a lot easier for you to see those morphologies. Maximum intensity projection makes it a lot harder to see those kind of things. So if morphology is important to you, changing the rendering mode can make it easier to present those morphologies. So in this particular case, I wanted to show you what that surface model looks like on its own, as it can be used as a representation of your data, so you can use it just for visualization purposes. Now, once we've created a surface object, we have a series of tabs that are available in that surface object. And the one that I want to show you right now is one that looks like a little plot or a graph. If I click on it, it will show me the statistics associated with the surface. So I explained this morning that a surface provides volumetric and morphological type data. So all the statistics are stored in the surface object. So I have currently 4 objects. I don't know how many you guys have. I'm not sure what threshold you use, but it said that it found 4. And the reason it found 4 is I have these other little objects in the back here, which are not part of my vessel, but they're being included in my count. So I'm going to show you a couple things. First of all, you can switch into select mode, and you can actually select your different surfaces. So either, the main vessel or these other structures. If you hold your command key down, control on a PC, you can select multiple. And you actually have an edit tab on here also. It looks like a little pencil icon. And this allows you to edit the surface. So we're going to just delete any extra stuff. What you're going to notice is if you go back to your statistics tab, it will automatically update your statistics with any changes that you make. So if you remove structures, it automatically removes those statistics from that table for you.
Sorry, do you have to go looking for the individual objects, or is there a tab that it could maybe light them up for you? Just if you have, let's say, a lot of...
It's a great question. So we're going to come back to that question because this is all to do with filtering and classification. So basically, when you start measuring structures, you have a lot of information about those structures. You know how big they are, you know where they are, you know how intense, what their intensity is. All of those parameters can be used to filter them, select them, remove them potentially. So I'll show you that in just a second. Okay. So one other tab I want to show you is one called Detailed. So by default, when you go into statistics, it shows you a count. The overall tab is the purpose for that is for counts. If you go into the Detailed tab, it gives you a list of objects that it has found. Now, I deleted all the other objects, so I should only have one statistic. Okay? What I do want to show you is everyone should see a statistic called area. That is the default because it's first in the list. That is your surface area. If I go into that little area option, you'll see there's a pull down list for all the other statistics that you can see or calculate. So if I scroll all the way down to the bottom, volume is the very last one. So the volume of this particular vessel is now calculated. Okay? So we're going to make it a little bit more challenging now because this is one object. Don't have to classify, don't have to do anything. So we're going to change the data set and look at something else, okay? So we're going to go back to the arena view, and when it asks you if you want to save what you've done, say no, unless you really did something amazing and you want to go back. So in this example, we're going to use now the plant cell data set. So it's red and green, and you should see it's one single cell, but it contains multiple chloroplasts. So it's this little icon or little thumbnail here. So if you double click, that will launch that into the surface view, and now we're going to make surfaces for the chloroplast. So we have a lot of things going on in this data set. There's a lot of chloroplasts. They're very close together, and they're at very different intensities as well. So it's a little bit more like what you're going to be facing when you're working with your own data. So we're going to create a surface. So we're going to click on the blue blob, and that is going to create a surface object. You don't have to rename it, but you can. So I'll call it chloro for chloroplasts. And we're going to go through the creation wizard, but we're going to use slightly different parameters this time, okay? So I'm not going to use a region of interest. These are demo images. They're tiny. We don't really need to worry about trying to conserve memory. So I'm going to go to the next step, and this is where we're going to choose the channel. I'm going to choose the green channel in this case. Now, I'm going to change from the default. So remember I said the default setting for thresholding was absolute intensity. I'm actually going to change it now to background subtraction. What a background subtraction threshold does is allow you to specify the size of the structures you're interested in so that it will apply a temporary background subtraction to the data so you can threshold that and see a more accurate segmentation of your structures. So remember, the absolute intensity allows you to have basically one cutoff for everything. So what the background subtraction does is basically remove the background to make it a lot easier for you to select anything else that remains. Now, the background subtraction is a temporary stage during this, so your data is not actually being changed permanently. So we're going to go in, choose background subtraction. It asks you for, I love this explanation, the diameter of the largest sphere which fits into the object. It doesn't really make a huge amount of sense, but let me explain it a different way. You want to basically provide the diameter of your smallest structure, okay? If you don't know how big your structures are, and a lot of people when they start analyzing data, they don't necessarily know, we have a little button in the top toolbar called Slice. If you click on Slice, that will take you into your slice view where you can look at the individual slices that make up that 3D volume. If you use the slider bar on the left-hand side, you can see you can pull that slice view up and down so you can look at different slices. So find a view where you can see a bunch of chloroplasts, and then click on the edge of one chloroplast and then click on the opposite edge, and you'll see it will draw a line for you. If you draw another, or if you click again, you'll see it will get rid of the previous line and allow you to draw another one. What we're doing there is we're making a very simple measurement, and the actual size of that line or length of that line is displayed over on the right hand side as a distance. Okay? So for those of you who are not familiar with how big chloroplasts are, they're about 4 microns. But for your data, you'll be able to go in and actually get a measurement for the structure you're trying to detect. So these are around 4 microns at their smallest diameter, so I'm going to enter in a 4 micron sphere. Okay, so that's the value I'm putting into background subtraction. Now I'm going to go to the next step. And what that does is allow me to minimize or basically remove the background temporarily so it's easier to see all of the information that is above background. Now, I want you to try something for me. You also have a little back arrow. So you have a forward arrow and a back arrow. The back arrow will take you back to the step previous, so you can keep going back and forth and fine-tuning your creation wizard. But I actually want you to try this with absolute intensity and tell me if you see a difference between those two modes. Okay? What you should see when you use absolute intensity is all of those chloroplasts look more fused together. Okay, they look like one bigger blob rather than being separated out. So the background subtraction allows you to get better and more accurate segmentation when you have variations of intensities. Okay?
How can we choose the surface detail?
Okay, that's a great question. Surface detail is a smoothing filter that is applied to the surface itself. And the reason we have that there is if you imagine your data set is actually made up of a series of tiny, tiny cubes. Okay? Those cubes are your voxels. Okay? So when you bring in your 2D slices and make a 3D volume, you have all these tiny little voxels that make up your data. And if you can imagine the very edge of your structures, your cells, your nuclei, your vesicles, actually have a very jagged edge. So I'm going to show you what this looks like. So if I go into the slice view and zoom up, you'll notice that these edges look really nice and smooth and rounded. Okay? That's because Imaris applies some interpolation to the display to make it look a little better. If I go into the preferences, there is an option under display called interpolate. If I turn it off and click OK, you're going to start seeing, in a minute, voxels or pixels. See these? Okay. That's not real. That's what we refer to as a detection artifact. That's what the detector picked up, but that's not the real edge of that structure. So what we do with the surface is we apply smoothing to basically make the surface smooth like the edge of that structure really should be. Okay? If we take smoothing off, you'll get a jagged edge. You can take smoothing off, and sometimes that's necessary if you're working with very, very small structures, because the value that we use, so I'm going to put the interpolation back on. Okay, and go back to this 3D view. The value that we use for surface detail is 2 pixels in diameter. That's the default value. Okay, so we apply a 2 pixel smooth. If your structure is around about 2 pixels, you may not actually get a surface on it at all. Okay, so there are cases, and the reason it's adjustable and you can also turn it on and off is for those cases where that smoothing may be too much. Okay, for the most part, that smoothing, that default value will probably be sufficient, but for people looking at puncta or very small structures, you have to be very careful with surface detail. That's something that you may actually want to turn off altogether. Does that make sense? Yeah, okay, perfect. Okay, so, oh, and one other thing, we display the surface detail in microns. So it's 2 pixels, but that's basically what that distance is in microns. A good rule of thumb is, if that value is close to the size of the structure you're trying to detect, that's a red flag, okay? So you want to turn that off if your structure size and your smoothing size are close together. If they're far apart, like your cell is 10 microns and that's 0.5, you probably don't need to worry. But if your structure is a micron and that's 0.5, that's something that you probably want to have to change. Okay. I think I just said want to have to change. Sorry, it's getting late in the day and I think my sentences are a little weird. Okay, so we're going to go now back to the threshold step. So I have background subtraction selected. I'm using a 4 micron seed size for that. And here is where, if necessary, we can adjust the threshold. There is one other feature I want to point out, and it's called split touching objects. Really useful for people who are imaging lots of different structures that are very close together. If they're close together and they touch, you will see a little fusion, you will see a little fusion in between those structures, okay? Those are actually 2 separate chloroplasts, but because they're literally bumped right side by side, the surface has made them one continuous structure. So this split touching object allows you to split those objects apart. So the same parameter we used a minute ago to define the smallest structure of interest for the background subtraction, I'm going to use the same parameter here. Okay, so I'm going to type in 4 microns and I'm going to go to the next step. And this is going to pull up a spot detection. Or not. Or not. Okay. This is where I'm going to give my laptop a break. It's been running all day with multiple applications, so I'm hoping it was just tired. So hopefully you guys didn't do what I just did. Are we good? No crashing? Yes. Okay. So let me get this back to where we were. We're doing this. We're doing 4 microns. Oops, cell wall. I need to change this. Okay, so we're enabling split touching objects. I'm using a value of 4, and now I'm going to the next step. Okay, so you guys should see something that looks like this. So every chloroplast should have a sphere that is 4 microns in diameter inside of it, okay? We have a quality filter which allows you to adjust a tolerance level, basically. So if you have structures that don't have a sphere or a spot inside of it, you can lower the quality filter, and that will basically place a sphere inside some of those dimmer structures, okay? So you can adjust the quality filter, but there's something I wanted to show you, and I found a really good one earlier. Here we go. So if you used a 4 micron sphere like I did, you will probably find a couple of instances where you have chloroplasts that don't have a sphere inside them, okay? And no matter what you do with the quality filter, it doesn't add in a sphere, okay? The reason for that is, in some instances, if you set your sphere size too large, it overlaps the boundary of the object next to it, so the software doesn't see that there's actually another peak intensity. So it ignores it. So the solution for this is reduce the size of the sphere slightly so you don't overlap the edge of one of those structures. So I'm going to go back and I'm going to type in 3.5 instead of 4. So I'm reducing the size of the sphere, and I'm going to go back, and you'll now see, hopefully, that you can put another spot. So basically, if you don't have a sphere or a spot inside of an object, it will not split it. It will continue to be one continuous structure. So you ideally would like to see a spot in every chloroplast. So once you've done this, you can now go to the next step, and this is where we apply a watershed filter to split our surfaces into separate structures. So what you should be able to see now is little cracks or breaks in your objects that define different structures rather than one continuous structure, okay? Now, the classification step is the last step in this process. And if you are working with a sample that has a bunch of stuff you don't care about as well as a bunch of stuff you do care about, and you're trying to figure out how do I pick up the stuff I want and remove the stuff I don't want, you need to pay attention to what makes it different. Is it its morphology? Is it its size? Is it in a position that's different? Is it brighter, dimmer, whatever? So this classification process allows you to fine-tune what you're looking for in the structures that are of interest. So if I go in, the default classifier is number of voxels. It's effectively an uncalibrated volume, but it's just a size filter. So you'll see down at the bottom here, you have a little histogram. That histogram is basically the x-axis is size, the y-axis is frequency. So it's just letting you see the distribution of sizes that you have in that surface. So if you click on the bottom on the left-hand side, you can remove or threshold out smaller structures. If you do the same thing with the right click on the top, you get rid of the bigger structures. So you've got control of an upper and a lower threshold for each classifier. Okay. Now, these classifiers are also additive, meaning that instead of just using a size threshold, you can specify a size threshold and then an intensity threshold, maybe a position threshold, but you can add them so that it has to meet certain criteria in order for it to get through that classification. So one that a lot of people find very useful, and I'm going to click on this add button at the top here, is one called distance to border. So for a lot of people who are counting cells, it should be somewhat straightforward to identify the cells of interest, but you tend to find everything all the way out to the very border of the image. So you may actually be seeing a partial cell, okay? So distance to border, so if I click on add and then go down to filter type, there is an option in that list called distance to border. If you click it, This will allow you to set a distance in microns from the edge of the image so you can actually specify what the exclusion range is around the entire image. Okay. Typically people use their cell size as an exclusion zone right around the edge. So if you know you're working with a 10 micron cell, you would use a 10 micron exclusion all the way around. Okay. And that just means that you are not counting or measuring half a cell or a portion of a cell. Okay. So just as an example, here's 10 microns, and you'll see it removes all of the structures that are within 10 microns. Okay, so classifiers can be really helpful when you want to make sure you don't pick something up in the first place, but I'm also going to show you one other feature. So I'm going to remove both of my filters in there for now, and I'm going to hit finish. Okay, so I now have a surface that contains multiple structures. So I'm going to turn my volume off, and you can see all of these various structures. If I go into the statistics tab, now instead of just getting one structure, I now have 261. And if I go into the detailed view, I now have an object by object measurement for all of the different statistics we calculate. Okay? So one other feature we have is something called a filter. So if you create a surface and then you want to start removing things, you can add a filter after the fact. So it looks like a little funnel. Okay, it's right next to the statistics tab. It looks like a little gray funnel, and it will allow you to add filters after the fact. So it's the same process, but you're doing it post surface creation rather than during the creation of a surface. And a lot of people use this one if they're trying to pull out particular populations. So let's imagine this was a field of cells. Some of them were green positive, some were red positive. So this kind of filtering would allow you to find all the cells, let's say with the nuclei, and then be able to go in and filter all of the green cells based on their intensity in the green channel. So these filters can be really helpful to help pull out populations within a surface that you've created. Okay, so in this example, if I wanted to do it based on intensity, I'm going to scroll down in here and look for mean intensity for channel 2, which is my chloroplast. Actually, I'll spice it up. I'll do mean intensity for channel 1. Channel 1 is a cell wall, so I'm going to look for all of those that are positive for red intensity. So you'll see that I can filter these structures based on whether or not they have any red intensity inside them. Okay? Once you've done that, there's a duplicate selection to new surface button right at the very bottom, and that will create a new surface that just has those that you filtered. Okay. Incidentally, if you were filtering with the purpose of getting rid of something, you could do the same thing. So if I go back into the filter tab, my filter tab is not behaving. Okay, I'll do it in this one. Neither of my filter tabs are behaving. Darn it. Okay, I'm going to walk you through this. Hopefully it's working for you. If you click on the filter tab and choose a filter to select some of your objects, you can then switch into the edit tab, which is right next to that, looks like a little pencil, and hit delete. So you have multiple options. You can either make a new selection or make a new surface with the ones that you care about, or you can select the ones you want to get rid of and go into edit to delete them. Okay? Okay. I'm going to show you a couple of other things while we're in here. First of all, if we go back to the statistics tab, I want to show you that we have the ability to export the statistics to Excel as an XLS or as a CSV file. So at the bottom of your statistics table, you will see little save disks. The single disk will save just the statistic that you're looking at, so in our case, area. If you go to the multi-disk icon, it will save all of the statistics that is calculated. Okay, so if you click on that icon, you'll see that you can choose Excel, CSV, or an XML as far as your export is concerned. Okay. Okay, one other thing I'm going to show you while we're in here. We have a ability to color code objects based on a statistic. So I talked about it this morning. It's called statistics color coding. And if I click on the little color wheel tab that brings me into the place where I can adjust or set up your statistics color coding. Oh. Can I help, or are you okay? Oh, my colleague is in the back. So if you need any help, she can totally help you out. Sorry. Okay, so I wanted to, so just to show you, we have this little option in here called Statistics Coded. You'll notice it will automatically color code your structures with a rainbow color table, and you can choose the statistic type that you want to use to color code them. So by default, it uses surface area, but you can go in and choose whatever you would like. So you could do it based on its volume. You could also do it based on where it is. So you could do position. And position's an interesting one because it does allow you to get an idea of where things are in reference to something else. So position information in this case is letting me look at where things are in reference to the X position or X axis. Okay, so that's where statistics color coding is. And just so that we're aware, there is also an option in there called base. Base will allow you to choose any color you want to color code your surface. So if you don't want to color code it based on the channel you created it with, you can color it whatever color you want. Okay, so we've now covered the different thresholding techniques for surfaces. The next thing that I want to go through with you is some animations. And then after we do animations, we're going to jump right into tracking. Okay? Does anyone have any questions on what we've covered so far?
I just have, if you could explain quick. We used the chloroplast, which were stained completely.
Yes.
How would you let's say you had a membrane marker of a cell, how could you just fill that space for the same?
So we have another model called Cell. And Cell is specifically designed to work with membrane labeled structures. So whether it was a lamin for nuclei or whether it was an actual membrane marker for the cell, you would need to use that model in order to pick it up. We're going to cover that in the next session.
Okay, perfect.
Okay, cool. So, animations. So we're going to do an animation portion, and then we'll jump into a different data set and look at tracking with spot detection. So animations. I'm going to go back to the volume view. I'm going to turn off my surfaces and look at the volume. In the bottom right-hand corner, we have an option called Reset, which actually takes you back to your original orientation of the data set where you're looking at the XY view. So if you click on Reset, it resets back and it fits it into the screen for you. Okay. To make it interesting, I'm probably going to put it into blend mode, and I'm probably going to change some of these values so that I can see the structure in a little bit more of a three-dimensional capacity. So I'm going to adjust the background. I'm going to vary the opacity here. I'm going to go into the chloroplast, make it look a little bit more solid, and then bring up the brightness. And I'm also going to adjust the background. So this is just kind of showing you how to incorporate different rendering into movies. So I'm using the blend mode to make it look more solid and three-dimensional. And now what I'm going to do is I'm going to bring in the animation component. So at the top of your screen, you're going to see a little video camera icon. It's pretty retro these days to see a video camera that looks like that. So I hope we keep it though. I hope it doesn't disappear. Anyway, so we've got a video camera icon. That is our animation tab, and it opens up the keyframe animation bar in the bottom of the screen. This keyframe animation bar is where we're going to create the animations. So the first thing you want to do is you want to set up the data to look exactly the way you want it to look in the movie. Okay, so you're basically you're setting it up to look how you want to start the movie. Now we have a blue rectangle at the bottom of the keyframe animation bar, and it has a series of vertical blue lines. Okay, those vertical blue lines are what we call animation frames, and they are what we're going to be using to create our animation. So first thing I'm going to do is I'm going to click on the Add button. And Add is going to add thicker bars, one at the start and one at the end of that animation. And these are storing the current configuration of the view that we're looking at. The zoom level, the rotation, the blend rendering, all of it, okay? Next thing I'm going to do is I'm going to rotate. So I'm going to switch into the navigate mode. I'm going to rotate, and I'm probably going to bring down the intensity a little bit. And I'm going to hit add. And then I'm probably going to rotate and look from this orientation. And I'm probably going to change my blend display a little bit here. Click add. And every time I'm making a change, I'm clicking the add button, and that is adding in these little thicker bars onto my animation frames. And basically what I'm doing is those vertical lines that were there initially are now becoming my transition frames. So from those keyframes that I'm adding in, I'm now adding in these transition in between. I'm going to zoom in now inside of my plant cell, and I'm going to click add. And you'll see that as I'm adding, you're just getting more and more of these thicker lines. Okay, now I'm going to go to my surface object, and I'm going to turn it on. I'm also going to turn my volume object off, and I'm going to click add. Okay, so not only is it remembering zoom and rotation, it's also remembering what objects are being visualized at a particular point. So this is something where you can decide you're going to present the original image data first and then start showing some of the models that you've used to detect the structures. Okay, so once I've done this add, I'm probably going to just flip it around like so. Click add. Now, while we're in this color tab for the surface, there's actually a little transparency slider bar in there. So even some of the settings for the objects themselves can be animated. So I'm turning the transparency to about 60%, and then I'm going to click add. And then I'm going to rotate it a little bit. And click add, and then I'm going to bring the transparency off. And click add. So this is where you can do whatever you want. So basically all the things, the changes that you're making, the rotations, the things you're adding, whatever you're doing, every time you do it, you click add, and that adds a keyframe. So if you now go back to click on the very first keyframe, so this thicker bar at the start here, it will take you back to the original position that you set. And if you click on the play button down at the bottom here, so the play button is on the keyframe animation, that will allow you to actually preview what your animation is going to look like. Okay? Now at this point, if there's things happening that you don't want to happen, or if there's things that you want to change, like me ramming into a chloroplast right there, you can go in and make changes and modify as you go along. So here's the transparency coming in, and then I think I flip it and then I turn the transparency off. So you can really look at what you've done and make changes. So let me pause this for a second. These keyframes are, when you add them, will automatically divide up the keyframes or the frames you have and space them equally apart. If you want something to happen quicker or slower, you can grab the keyframe and drag it to a particular location. So for example, if I wanted this rotation to happen quicker, you can grab these two and bring them closer together, and that rotation will then happen faster. So if I click play, so here we're kind of going slowly, it's going to hit the keyframe, and then that last transition will happen a little faster. I chose one that didn't really do much, but okay. So you can move them closer and further apart based on what it is you're trying to present. So all of that is being remembered. Now one other thing I want to show you. The more keyframes you add, the more animation frames you will need, because the more keyframes you have, these animation frames, these thinner animation frames in between, will start getting divided up by all your keyframes, so the transitions will end up being faster. So if you want more smooth transitions, you're going to have to increase the number of animations you have, the animation frames you have. So all you need to do, the default number incidentally is 200 frames. You can just increase it. So if you make it 300, you'll see that it will just interpolate and add more for you. Okay? Now, one other thing I wanted to show you. If you play through the movie and something happens and you don't like what it did, what you can do is click on that keyframe, make the change, and click modify. Okay, so maybe you zoomed in too far, or maybe you didn't have it centered. You can go to that keyframe, make the change, and then click modify. So everything you do is also modifiable. Also, if there's a keyframe that you added in and you just don't need it or don't want it, you can also hit delete. Okay? Now, I will tell you, animations are probably where you're going to spend most of your life. People tend to get carried away and super pernickety with how their animation looks. So this is an area where the more time you spend with it, the more tricks you're going to learn, and you'll get better and more familiar with it. Okay, so I want to show you how to save. Now there's one thing I forgot to mention, and I talked about it in the last session, so I'll talk about it here too. So I talked about something called a clipping plane this morning, where you can basically hide and show objects based on where they are in the data. The clipping plane icon looks like a pair of scissors. So it's at the top of the scene list. If you click on the scissor icon, it will add it into your view. And if you rotate, you'll see that you've got this white manipulator that is hiding some of your data and showing some of your data. Okay? In the clipping plane configuration, so if it's selected, over here you're going to see an option called flip, so you can change what direction is being hidden versus shown. You can also choose an orientation at which you want to clip the plane. Okay? But you also can manipulate that on your own too. So the manipulator is made up of a thicker part and a thinner part. The thicker part allows you to adjust the depth of that clipping plane, and the thinner part allows you to change the orientation of the clipping plane. So the two of them together can help you to figure out how you want to slice through that particular volume. Okay. In a lot of cases, when you're imaging something on a microscope, whatever you're imaging may not be oriented exactly the way you want it to be, so this is something that can help you kind of resection and include in movies. Okay, so one other thing that's important is the position of the clipping plane impacts what is clipped. So you'll notice that right now the clipping plane got put in right under your light source. That means everything underneath the clipping plane is being clipped. So as I use that manipulator, the frame's going, the data's going, everything's going. If you want to only clip specific things, make sure the things you don't want to clip are above the clipping plane in the list. So you can grab objects and put them above, or you can move the clipping plane down. Okay, so an example of this is, let's say your surface is here. So I have the data on, the surface is on, I also have a clipping plane, but the volume is above the clipping plane, the surface is below. So if I go into that clipping plane and now move it back and forth, you'll see the only thing it's removing right now is the surface. If I change this and put the surface above and the volume below, it will change what is being clipped.
Can you have multiple?
Yes, you can.
Okay.
Yeah. That gets confusing, but yes, you can. So the question was, can you have multiple clipping planes? You absolutely can. So you can clip it from multiple directions. You can clip it from top and bottom. You can do all sorts of different things. Like I said, animations can become a huge time sink because you can do a lot of cool things. So you tend to, the more you add, the more complex it gets. Okay, so this can also be incorporated into your movie. So I'm going to add this in at some point. Let's see. Okay, so I think I'm going to add it in for me towards the end. So I'm going to add in my volume. I'm going to put my clipping plane in here. I'm going to change it to do an XY, or maybe I'll do another plane. And I'm going to pull this all the way back. I'm going to click, okay. I'm going to pull it all the way like this. Click add. And now I'm going to do a progression through the clipping plane by dragging it through. And then I'm going to click add. And now I'm going to turn the surface object off. Okay, so hopefully, if I now play this, I should have started a little bit closer to the clipping plane, but now I just do my rotation with the surfaces on. In a moment, I'm going to add the volume object back in, but I'm bringing it in with a clipping plane. And then I'll turn the surfaces off to see the, I think in the next frame. Okay, so you can incorporate a lot of those tools in a particular way. So whatever, oh, and then also it's clipped at the end here. So you can incorporate this in lots of different ways to try and do different things and present different aspects of your data. Okay, so the last thing I'm going to do is I'm going to delete what we did at the end there. You can also save, obviously, and export the movie. So there's a little record button. That record button will give you, turn that off, will give you the option of choosing from a couple of different formats. So we have MP4, we have AVI, and we have a regular TIFF. Now, we have simplified that somewhat because most people using Imaris are using this H.264 codec currently, because it gives you a nice compromise between resolution and size. So this one is one that a lot of people are using for 3D movies. The TIFF option is if you want to take the data elsewhere and use a different codec. Maybe you're using iMovie or something else. And then there's also a raw AVI, which is uncompressed, so it'll be very, very large. So in this case, you choose your codec or your compression, and then you choose the frames per second, and then you hit save, and it creates a movie for you. I'll do 10 for now. Hit save. Okay. I have one from the last session we did. Okay. It will play through just to verify that this is how your movie is going to look. And then once it's finished creating the movie, it will pop up with a little movie screen. A little tip for you also, if you hit the record button again while it is saving, it will actually save everything up until that point. So it doesn't stop creating because it's already created those frames. You just get a portion of the movie rather than the entire thing. So hitting the record button partially the way through will just give you a portion of the movie. Okay. Any questions, or are we good? Good? Okay. So we're going to move on to the next stage, and that is tracking. We didn't do it in the last session, so I promised myself that I would do it for this one. So I think we have about another 15 minutes. Is that right? 15? Yes. Okay. So if everyone could open up a data set that's labeled R18 demo, it's a single channel image, but it is a 4D image. So it's a live cell data set. We're using pretty simple data sets for the purpose of tutorials so you can understand, but also because you're using these little iMacs, I don't want you to crash the system with massive data. So we're using some small data sets for this. Now, I'm going to use a different model this time. We've done quite a bit with surfaces so far, so we're going to introduce spots. Okay? So we're going to use spots to identify the location of every single one of these cells, and we're going to track those cells over time. Okay? So, first thing that we're going to do, I just want to show you down at the very bottom, when you bring in a time-lapse data set, you're going to notice that you have a time bar at the bottom. And that time bar can be interacted with, so you can grab the little red triangle and grab it back and forth, and you can see that in doing that you can move through your time points. You also have a play button, so it will actually play through those time points for you. There is a little settings button that looks like a little tool, which will allow you to set the playback rate, whether it's a loop, whether it's back and forth, what the speed of that playback is. So that can all be configured in the bottom here. Now, what I'd like to do is I would like to identify the location of each of these T cells. I don't think I mentioned these are T cells. And they're all over the place in 3D, so I want to identify them automatically in every single time point. Okay? So the first thing I'm going to do is I'm going to jump into the slice view, and I want to get an idea of how big these cells are. So I'm going to do what we were doing previously. Draw my little line. It comes up as about 6.8 microns, about 7 microns. I'm going to measure a couple of other ones just to get a sense of how big these are. So you want like an average size. Okay, so I'm probably going to go with about 6 microns for these. And so what we're going to do is, instead of using a surface, this time we're going to use the spots object, which is right next to it on the left-hand side. It looks like little orange spots. Click on it, and it will add a spot object in. So I'm going to call these T cells. And we're going to go through the creation wizard. Now, we've done a creation wizard for surfaces, and the concept is exactly the same for all of the different models we have. The parameters you're entering might be slightly different, but the concept is you're going to go through a set of steps to figure out what best identifies your structures. So in this one, it automatically assumes you're going to be tracking because it's a 4D data set. Okay, so tracking is turned on. I do want to show you one thing with a region of interest. A region of interest for a time-lapse data set is not just X, Y, Z. It's also a time region. So let's imagine you have a data set where the cell died. You don't want to track the entire thing even though it died. You want to get the portion where it was alive and still doing something. So if I select this segment region of interest and go to the next step, you'll see that there is a 4th dimension there called time. So you can specify the range of time points that you want to measure. Okay. So I think I have about 30 frames. So in this case, I'm probably going to do all of them, but I wanted to show you that this was here because a lot of people want to only track a portion of their data. Okay. So I'm going to go back a step and turn it off. I just wanted to show you that it was there. And we'll now go to the next step. So because I had that region of interest now deselected, it misses that step altogether, or bypasses. So here we choose the channel, and we also enter in the estimated diameter for the cell. So in this case, whatever you decide you want to use, I think I'm going to use about 6 to identify my objects. Notice there's another little checkbox right underneath. Again, it's another one of those non-intuitive explanations. It's called a model PSF elongation along z-axis. What that really means is, if you look at your structure and turn it and look in Z and it looks elongated, which a lot of structures do, it's a spherical aberration, it's an inherent issue with a lot of optics, you can basically make the spot look like an ellipsoid rather than a sphere. And that is, the purpose of that is so that instead of potentially finding multiple spots in one object because it's elongated, you're filling that whole structure with an ellipsoid. Okay, so I'm going to show you what this looks like. So just to give you an example, if I go into the slice view, we have another option called section. And if I look at one of these T cells, here I'm looking at the XY. This is the the YZ, and this is the XZ. And you can already see that this round structure looks kind of elongated in the Z-axis. That's somewhat normal. So the section view gives you the ability to measure that structure in all 3 dimensions. So I'm going in and not only measuring that in X and Y, I'm also measuring it in Z as well. Okay? So it gives me a measurement of about 5.7. I've got about 4 microns, which is kind of small. I think that was, yeah. So it's about 6 by 6 by 10. Okay? So I'm going to go back to my 3D view, and I'm actually going to turn on that Z-axis elongation, and I'm going to switch that to 10. Okay? I'm now going to go to the next step, and what it's going to, and now it's going to place a sphere in most of the cells, usually the brightest cells. But what I want you to see is if you use that Z elongation, when you rotate it and look in the Z-axis, they look like eggs rather than looking like spheres. So it's just a better representation of what your data looks like, and it will be more accurate, especially if you have a significant amount of elongation. Okay. One other thing I want to tell you at this point is you have a quality filter. This quality filter allows you to incorporate dimmer structures that were not automatically picked up. So if you see cells that should be tracked and are not being picked up, you can add a filter, or sorry, you can reduce the quality filter to pick them up. Now I have quite a bit of background in here as well, so we don't want to confuse cells with background. So I'm just looking to make sure that as I move through time, that I have a sphere or spot in each of my objects. If I need to, I can reduce the quality, but for the most part, it looks like it's found most of my cells. Okay? Yeah. Yes, you can. Yeah. So the next step, if I click on the next button, is your manual intervention. So if you want to add a spot or remove a spot, you can do so here. So this is called the edit spot section. So what you can do, and let me show you this, you need to be, you need to zoom up, you need to be in select mode, and you'll notice your cursor will have a little box that follows it around. That box is the same size as the spot you entered. So it's a 6 micron diameter and then 10 in Z. So this little box that we're looking at is representative of the sphere size we already have. So if you want to add a spot, let's say we want to add one right here, you put that box over the spot, hold the shift key down and click on it, and it will add a spot in that location for you. Same to remove a spot. So if you hold this, so put your box over the spot that you want to remove. So I will do it with this one right here that I added, shift, select, remove. Okay? Now I will tell you, if you have a long time lapse, the last thing you're going to want to do is sit and place spots manually. So a lot of what you're trying to do with this is have the software understand what you're looking for. So if it's not finding things, there's a reason it's not finding things. It could be that it needs some background subtraction. It could be that your sphere size is too big or too small. So there's other reasons that could mean that you're having to go in and manually place things. So if at all possible, the better you set up your criteria and the better you process your data prior to, the more accurate this will be in the long run. Okay, so the next thing I want to show you is if we go to the next step, we get to the tracking algorithm. Now, Imaris provides 5 algorithms. We actually have a total of 6, but you won't hear about that unless you're coming to the afternoon session. So we have 5 in this dialogue. Some of these you will probably never use. I will talk to you a little bit about the ones that you are more likely to use. So the default one is called autoregressive motion. There's also one in there called Brownian motion, and there's one in there called lineage. Those are the 3 that you are more than likely going to be using. Now, autoregressive and Brownian motion assume there's no cell division. So if you're doing cell division, if you're looking and tracking cell division, the only one you should be using is lineage. If, however, you're looking at migration, or if you are looking at just overall motility of your cells, you're going to have to make a decision as to whether you're going to use autoregressive or Brownian. And this is actually an easy one. If your cells are migrating in a particular direction, whether they're meandering there or going straight there, if you can visualize that movement and it's always in a forward momentum, you're going to use autoregressive motion. If your cells are moving randomly, or if sometimes your cell moves forward and then backward, you're going to use Brownian motion. And if you just don't know, use Brownian motion. Okay? The difference between the two, autoregressive and Brownian, autoregressive makes an assumption that we know where the cell is going based on where it last was. So if it moved from A to B, we're kind of making the assumption it's going to keep moving in that direction. Okay, so our search area is focusing 180 degrees based on that last movement that it made. Okay, so if a cell moves forward and then it moves back, autoregressive motion will not track that. It's only going to move it if it's moving in a forward momentum. Okay, so Brownian motion, I always say to people, if you're not sure, use Brownian because it looks 360 degrees around for the next connection. Okay? There's no assumption of where it's going to move. It looks all the way around. Now, that distance at which it searches, so in this one I'm going to use Brownian motion. The distance in which it searches for the next cell or the connection is based on this max distance parameter. Okay? So if you have a cell that is moving, 10 microns in each time point, you're going to want to have at least a search radius of 10 microns so you can find it in the next time point. What you have to be really careful about with your tracking parameters is most people will be like, oh, I'm just going to throw 100 microns in there and then I'll get everything. The problem is if you make that search region too big, you start to incorporate other cells and you could have false connections. So you really need to find that optimum distance that is not too short, because if it's too short, it won't find it. If it's too large, you'll get false connections. So you need to find that perfect distance. It doesn't have to be perfect. You need to find that distance that accommodates the movement. Now, temporal resolution is a big part of this. Remember this morning, if you were there this morning, I talked about if you can follow it by eye and know where it's going by eye, that means you can track it. If you look at something and you cannot tell from one time point to the next which cell is which, you don't have enough temporal resolution. And so that's something that I think a lot of people are like, I don't know what's going on, but Imaris can figure it out. If you can't figure it out, the chances are no software is going to figure that out for you. Okay? So temporal resolution is important. And ideally, you don't want to have time points too far apart. So you ideally want to have it be no more than about a cell diameter movement between adjacent time points. That's a perfect world scenario. And I know with live cell, there's a lot of problems and a lot of things you have to worry about, including your photo bleaching and keeping them alive and all that other stuff. But ideal world would be no more than a cell diameter. Okay? So in order to figure out what your movement or your distance is, go back to your sliced view. And you can place a little spot in the center of one of your cells, go to an adjacent time point, and measure the movement. In this case, that one barely moved. So you're looking for one of your faster moving cells. So let's say at we don't want to measure that distance. Okay, so I'm going to go here, and then I'm going to go to another time point and put a value in there, and that was a distance of 5 microns. So you want to look, though, for your fastest moving cells. Don't do it with a cell that's not moving. Look at the ones that are moving and figure out what, between adjacent time points, how far could it move. So with this data set, I'm probably going to go with about 20 microns because I do have one that moves pretty fast. So I'm going to pump this number up to 20. The gap size is the number of time points your structure can go undetected. And if it comes back in, it will continue tracking it. This one you have to be really careful about. A lot of people are like, oh, it just popped off the edge and it came back in, it's the same cell. You don't know if it's the same cell. If it goes out of focus and comes back in, do you really know it's the same cell? I guess there's assumptions you can make, but this one, you don't want to have that number be too large. Typically anywhere between 0 and 3 is a good number. But this is effectively the number of time points that a structure can be missing and still be tracked if it comes back in. Okay? So I'm going to go ahead and go to the next step. And here we have the tracks. The tracking component is super duper fast. The part that takes time typically is the object detection. The tracking part is really fast. And what we're looking at right now is the entire track from start to finish. And the color that we see represents where it began and where it ended, because some tracks start later on in the experiment, some disappear early in the experiment. So the color coding is an indication as to how well a structure or how long that structure was really in your experiment for. Okay? So the blue is the beginning and the red is the end. Now, we also have one last filter, and the last filter is for your track statistics. So you may have hundreds of cells, but all you really care about are the ones that are moving quickly or the ones that aren't moving at all. So you can set up a classification based on those tracking statistics. So just to give you an example, the default one is track duration. So you can specify, I'm only interested in tracks that are there for at least a certain length of time. You can also specify track length, or you can also specify the speed at which the object is moving. There are a number of different statistics that you can use. Okay? In this case, I'm going to leave it as is and hit finish. And what we have now is little spheres representing each of the cells, and then we have the little trajectories that actually represent the movement of each of those cells. Okay? And if I hit the play button, you will be able to see the spheres representing your cells moving along those trajectories. Okay? Now, I do want to show you a couple of things. First of all, those spheres themselves can kind of obscure your view of the cells. So the first thing I wanted to show you is in that very first tab that looks like the spots icon, you can change the sphere into a center point. So it shows you just the centroid of the structure. So you can see the original data and then see it moving along your tracks. You can also turn it off altogether, but this is just a good way for you to know whereabouts in the track you are. We also have a feature that has been around for a long time, but we just made a big change to it. So it's something called a drag and tail. And a drag and tail, instead of seeing the track in its entirety, you can see where it has been. Okay, so you see the last however many seconds of your experiment, you can see the tail of that track. Now, what we have changed is you can now also see what's coming in the future. So you can see the track. So for every time point you're looking at, you can look a certain distance into the future as to where it's going, as well as where it's been in the past. And you get to specify that in seconds. Okay? So right now I have drag and tail on, so instead of seeing the entire track, I'm just seeing it where it was for the last 11 seconds. And then I can also specify that I want to see 11 seconds into the future as well, and that will show you both, 11 seconds before and after. Okay? This is really good for movies. If you put all of your tracks on at the same time, you'll tend to find it will look like a ball of yarn in a lot of cases, and people can't really see what's going on. The drag and tail really helps to allow people to follow where the cells are going. Okay? So drag and tail is really useful. The other thing I wanted to show you, and this is one that doesn't get used very often, but I'm going to point it out. We have something called Tracks Displacement Display. It's down at the bottom, it's a little checkbox, and all it does is add an arrow. The arrow shows you, it's basically a connection between the start of the track and the end of the track. And it's very useful if you are looking at cell migration, for example, because you get all of these arrows that help you to decipher where the overall movement of your structures are. So just wanted to show you that that is there. And remember, it's a straight line between the beginning and the end of that track. Okay. There is a huge number of statistics, which I'll come to in a second. I also want to show you that we have a track editor. No tracking algorithm is going to be perfect, and that's probably because no data set is perfect. So there's always going to be situations where cells were dying, something happened, so you may need to kind of intervene and do some things. So what I want to show you is if you click on the icon that looks like a little marker pen, and you can stretch this out a little bit to see it better. But what we have here is a display of all of your tracks. And these tracks, I'm just looking at my time right now and I'm already over, so I apologize. But each one of these lines represents a track, so you can see where they start and where they end. You can also see the number of tracks that you have. And one thing that I want to show you is if you ever need to modify a track, so I'm going to put the spots back on to see these a little bit better. If you ever want to modify a track, you can click on the object. You'll see it will select the track that pertains to. And there's an option here called Rebuild Around Selection. So it means that it will put all of the tracks that are close to the one you're interested in into this bar. So in my case, I don't have that many tracks, but if you had hundreds of them, it would allow you to see all of those that are 15 of those that are closest to it. So it makes it a little bit easier for modification purposes. But let me show you. So the way that we would connect a track that is broken, and I'm going to look for one that I want to connect. So I'm going to use this one here because I can't really see anything else that I would want to connect. Okay. So what we're going to do is I'm going to select a track that I want to connect. And I'm going to hold down the command key for you guys, control for me on PC, and select those 2 tracks. And up at the top here, I have an option of what I want to do with those tracks. So this option right here, if I put my mouse cursor on it, will allow me to connect those 2 tracks together. So if I click on that option, those 2 tracks will get connected and all the statistics get updated to connect those 2. I also have an option to completely break all of the tracks. And it could be that there's one track that just was a complete mess and you want to just take all of the track off because you're going to connect it all manually. So this one here deselects all of the tracks. And then we also have an option here if you do want to connect 2 tracks that should be the same track. You would select 2 options and you'll see that this option here will allow you to take the start of or the end of one track and connect it to the start of another track. So all of these can be done either by selecting these objects or these tracks in your track editor, or you can select them over here and make the necessary adjustments. Okay. Now, one last thing, and I did not get to go into it at all, we have a drift correction option. So if your object is moving in time and it's an external movement, maybe there's a vibration or a drift or, you know, something else, like maybe it's an embryo and it's rotating, you can add in a correction for that. And in this dialog, we have a little button called Correct Drift. The idea is if you want to use it in this dialog, you would track something that was stationary. Maybe there was a piece of debris or something you can use as a reference for something that's not moving. You can track it, select its track, and then hit this Correct Drift button. And this will come up and give you the options for either translational drift adjustment or translational and rotational drift adjustment. But it will actually change the data to fix that correction, that drift or that movement, so that your tracking statistics will be corrected. Okay? Okay, so I'm sorry, I have my last group coming in for the day, so I'm going to finish here. I will have this recording for you so you can look at it later. I'm also going to ask if there's any kind of last minute questions. No? Oh.
I don't know how to add sport.
How to
add sport when I label sport and then we connect as a tracker, but in the maybe first 2 steps we can add or delete.
So I'm going to come and help you in just 1 second, okay? So if you have any questions, this recordings will be with Jordan and Adam in the BIDC, so you can go and ask them. I don't know how you're going to distribute, but I'll let you deal with that. And then if you do need any other resources, go to our website, bitplane.com, go to the Learning Center. We have a bunch of video tutorials in there that will help to go through specific things. It's also searchable, so you could type in tracking or you can type in colloq or whatever it is you want to learn about, and it will open up all of those video tutorials for you. Okay, so thank you very much. Good luck. It's available in the BIDC if you want to start using it, and I hope you enjoy it. Thank you.
Do we have time for a break if we're in the next session as well?
Hopefully.